Effect of plant extract on mesenchymal stem cell proliferation and differentiation. By Bhuvan Saud, 1, 2 Rajni Malla, 1 and Kanfi Shrestha. Stem cell has immense potential in regenerative cellular therapy. Mesenchymal stem cells, MSCs, can become a potential attractive candidate for therapy due to its remarkable ability of self-renewal and differentiation into 3 lineages, i.e., ectoderm, mesoderm, and endoderm. Stem cell holds tremendous promises in the field of tissue regeneration and transplantation for disease treatments. Globally, medicinal plants are being used for the treatment and prevention of a variety of diseases. Phytochemicals like naringin, icariin, genistein, and resveratrol obtained from plants have been extensively used in traditional medicine for centuries. Certain bioactive compounds from plants increase the rate of tissue regeneration, differentiation, and immunomodulation. Several studies show that bioactive compounds from plants have a specific role, bioactive mediator, in regulating the rate of cell division and differentiation through complex signal pathways like BMP2, RUNX2, and WNT. The use of plant bioactive phytochemicals may also become promising in treating diseases like osteoporosis, neurodegenerative disorders, and other tissue degenerative disorders. Thus, the present review article is aimed at highlighting the roles and consequences of plant extracts on MSCs, proliferation and desired lineage differentiations. Stem cells are precursor biological cells that have the ability to self-renew and differentiate into multiple mature cells. 1. Stem cells divide into 2 major categories, i.e., embryonic stem cells and adult stem cells. Depending upon the differentiation capacity, they can be classified into unipotent, multipotent, pluripotent, or totipotent stem cells. These cells provide the platform to investigate cellular development, maintenance, and differentiation. From the initial development of human civilization, plants have been used as a medicine for improving growth and development. Medicinal plants are widely acceptable for the treatment of a variety of diseases. The World Health Organization, WHO, declared that the best sources of a variety of drugs are plant derivatives. Globally, around 75% of the population from the developing and developed nations like Britain, Germany, and France use plants and their extracts as a medicine. Out of 150,000 plants being studied, medclinically important components have been observed in many of them. Plant derivatives have shown to promote stem cell proliferation and multilineage differentiation. The bioactive compounds obtained from plant extracts could become an alternative, cost-effective treatment for bone marrow transplantation and cancer. Most plants have been used both traditionally and therapeutically, but the exact mechanism of action on MSCs of only few plant extracts has been proved. Establishing the differentiation of MSCs into desired lineage-committed progenitors in the presence of a certain plant extract can open a new horizon for regenerative medicine and treatment. Thus, the present review highlights the role of bioactive compounds from plant extracts on MSCs proliferation and differentiation and their use in regenerative therapy and medicine. MSCs are divided by mitosis but are not capable of unlimited cell division in vitro due to senescence, also called irreversible growth arrest phenomenon, first described by Hayflick in the 1960s. According to ISCT criteria, MSCs must be able to differentiate into multilineage cells including osteoblasts, adipocytes, and chondroblasts, but it depends upon in vitro conditions as well as the cell source. Depending upon the source, MSCs have high potential to differentiate into osteoblast, chondrocyte, adipocyte, skeletal muscle cells, endothelial cells, cardiomyocyte-like cells, and neuronal cells. MSCs differentiate into osteoblast, chondrocyte, adipocyte, tenocyte, and vascular smooth muscle cells. In addition, periosteum MSCs, PMSCs, synovial MSCs, SMSCs, adipose tissue MSCs, MSCs, circulating MSCs, CMSCs, and tendon-derived MSCs, TDMSCs, also have potential of multilineage differentiation under in vitro standard condition. Effect of medicinal plant extracts on MSCs. Globally, plants and their products are used for improving health. Plants have been providing endless sources of medicine throughout history. Their method of production, purpose, and method of use vary. The USA has categorized plants into dietary supplements, intended to supplement the diet and usually consist of vitamins and minerals, drugs, over-the-counter drugs, and botanical drugs, complex extracts used for treatment. Extracts from different parts of a plant, root, bark, flower, leaf, and seed, may be used for different therapeutic purposes. Ayurveda, Southeast and Middle East Asian, and Chinese traditional medicines are the roots for use of natural products in treating diseases. Plant extracts contain bioactive compounds like polyphenols, flavonoids, and many other compounds and chemical substances which play important roles to treat both communicable and non-communicable diseases. Due to health benefits, phytochemicals from plants generate a lot of interest, demanding further scientific evaluation. According to the National Institutes of Health, USA database, of the 680 clinical trials on MSCs, 27 have used dietary supplements including herbal compounds, natural compounds isolated from blueberry, green tea, catechin, carnosine, and vitamin D. 3. Have shown to promote the proliferation of stem cell of bone marrow. Dietary fatty acids, oleic acid and linoleic acid, promote the proliferation of hemopoietic stem cells. Under standard in vitro condition, supplementing plant extract may induce increased rates of MSCs proliferation and multilineage differentiation. Moreover, studies have shown that extracts also increase pluripotent stem cell proliferation and anti-cancer potency. Proliferation and differentiation stimulants. Medicinal plants and herbs have always been valuable in disease treatment. Recently, researchers have investigated and identified those pharmacologically active substances which are responsible for disease prevention and treatment. Recently, medicinal plants have received considerable attention as stimulants for stem cell proliferation in vivo and in vitro. In vitro studies of natural bioactive compounds have suggested that plant-derived substances enhance the adult stem cell proliferation and on the other hand inhibit the proliferation of cancer cells. Several studies have suggested that the proliferation ability of MSCs is influenced by the dose of the stimulant compound, where higher doses of cellular toxicity appear. Using 1 to 100 μg/ml extract from a citrus increased the human MSCs proliferation and osteogenic differentiation, while using 200 μg/ml concentration decreases MSCs growth. In rat MSCs, naringin 50 μg/ml concentration increased growth of MSCs and a higher concentration at 100 μg/ml suppressed the rate of proliferation. In addition, extracts from brown algae Laminaria japonica, fucoidan, enhance the proliferation of human-derived MSCs when using 0.1 to 10 μg/ml concentration. Studies have shown MSCs differentiation into osteogenic, neurogenic, and endothelial/vascular progenitor cells in the presence of plant extract supplements. Certain phytochemicals may increase the cellular proliferation and at the same time reduce the time required. The effects of plant extracts on MSCs. Differentiation and their possible mechanism are shown below. Effect of plant extract on MSC proliferation. Glycine max var. Vegetable soy peptides, 25% and 20% increase cell proliferation rate, and TGF. Beta-1 plays a crucial role to induce proliferation. Ocimum basilicum induces MSC proliferation and reduces doubling time, dt, at 10 μg/ml. Concentration. Glycyrrhiza glabra, licorice root, increases significant level of proliferation at concentration 10. 50 μg/ml. Apple proliferation promotes by ERK-dependent cytokine production. Ginkgo biloba 25 mg/L increases the cell proliferation by 30%. Effect of plant extracts on MSC differentiation. Ginkgo biloba. Osteogenic. Increases transcriptional levels of bone morphogenetic protein 4, BMP4, runt-related. Transcription factor 2, RUNX2, beta-catenin, and cyclin D1. Olea europaea leaf. Endothelial/vascular. Argenesis. Increases gene expression for vascular endothelial growth factor, platelet. Derived growth factor receptor, and vascular endothelial growth factor receptor. VEGFR-1. Ocimum basilicum. Osteogenic osteonectin and osteocalcin levels increase. Glycyrrhiza glabra, licorice root. Osteogenic osteocalcin, RUNX2, BMP2, and ALP gene expression upregulate. Phytochemical compounds. Naringin. Good sources of naringin include lemons, grapefruits, pomelos, blood oranges, tangerines and tangelos, grapes. The zest or colored rind of citrus fruits contains higher concentrations of both vitamins and flavonoids. In the genus Citrus, the most common bioflavonoids are naringin and hesperidin. They occur mostly in the peel of some citrus such as Seville bitter orange, sweet orange, grapefruit, lemon, although they may also be present to a lesser extent in their pulps, juices, leaves, seeds and flowers. Naringin, 4,5,7-trihydroxyflavanone 7-rhamnoglucoside, is an abundant flavone glycoside present in citrus fruits with interesting biological and pharmacological actions. The name naringin is probably derived from the Sanskrit term naringi meaning orange. Naringin is a flavonoid glycoside that is abundantly contained in the skin of grapefruit, also called pomelo and orange, and is the origin of their bitterness. Naringin, a flavonone glycoside, occurs naturally in the pericarp of citrus fruit, and particularly of grapefruit, Citrus paradisii, where it is the predominant flavonoid found and is responsible for the bitter taste associated with the fruit. Citrus plants are a good source of flavonoids. Naringin, naringenin, nobelidin, narirutin, and hesperidin are the most important flavonoids thus far isolated from citrus fruits. 13. These flavonoids were found to possess strong antioxidant and anti-inflammatory activities both in vitro and in vivo. Many of the bioactivities of citrus flavonoids appear to affect vascular endothelial cells. Naringin, naringenin 7-onea hesperidas, belongs to the flavonoid group, has an antioxidant effect, is anticancerous, and is used for reducing the cholesterol level. It is also used for the treatment of bone disorders like osteoporosis and osteoarthritis. Naringin has a potential to induce prosteogenic effects which could promote the proliferation of stem cell. In vitro condition, it has shown to enhance the osteogenic differentiation by increasing the expression of RUNX2, OXS, OCN, and COL1. And increase the proliferation by activating the ERK signaling pathway on human MSCs. In rat BMSCs, naringin increases the mRNA levels of osteogenic genes and Notch1 expression. In human amniotic fluid-derived stem cells, HAFSCS, naringin promotes osteogenesis via BMP and WNT-beta catenin signaling pathways. In addition, it increases the expression of bone morphogenetic protein 4, BMP4, Runt-related transcription factor 2, RUNX2, beta-catenin, and cyclin D1 in a dose-dependent manner by 1 to 100 μg/ml. At 1 μm concentration, it promotes the proliferation and differentiation of human periodontal ligament stem cells, HPDLSCS, both in vitro and in vivo. The proliferation and differentiation are dependent on the dose of naringin in dog-originated MSCs. Rhizoma. Dry naringin is used commonly in the treatment of osteoporosis and bone non-union in traditional Chinese medicine. The flavonone may become a potential therapeutic candidate to promote the osteogenesis. Genistein. Genistein has structural similarity to human estrogen, so it is also called phytoestrogen. It is one of the most abundant isoflavones in soy. Isoflavones belong to the group of flavonoids, and they act as phytoestrogens, antioxidants, and anti-cancer agents. Genistein, when added to medium, 10,7-M and 10,8-M, promotes bone formation and also increases the level of alkaline phosphatase activity and DNA content. Genistein promotes the HBMSCS, human BMSCS, to osteogenic differentiation through an ER-dependent mechanism. Also, BMP-dependent SMADs and RUNX2 signaling play important roles in the process. In addition, it could stimulate differentiation through the P38 MAPK-SPA1 pathway. However, studies have shown that it also induces adipogenic differentiation, promotes triglycerides activity in HbMSC, and suppresses osteogenic potential by upregulating the expression of PPAY-gamma. An in vitro study shows that genistein stimulates HMSC, induced cellular proliferation and survival of cells, and enhances anti-apoptotic capacity. Resveratrol. Resveratrol, RSVL, is a natural type of polyphenolic phytoestrogen. RSVL is mainly found in red grapes, blueberries, peanuts, and other plants. The effect of RSVL on stem cell is well documented. It enhances the HbMSC proliferation and potential to differentiate into osteocyte by activation of extracellular signal-regulated kinase 1/2, ERK1/2, and P38 mitogen-activated protein kinase MAPK, signaling through an ER-dependent mechanism. RSVL showed the effect on HMSCs in dose and time-dependent manners for the self-proliferation and differentiation. 0.1 μm RSVL promotes cell proliferation, but 5 μm or above inhibits cell self-renewal by increasing the senescence rate and cell cycle arrest in S phage. It also helps MSC differentiation into osteogenic cells and suppresses differentiation into the adipogenic lineage. Resveratrol enhances osteogenic differentiation by upregulating HMSC mediated through the SIRT1/FOXO3A. It activates and enhances the proteins SIRT1 and FOXO3A, respectively, in an independent manner. Resveratrol also promotes osteogenesis by upregulating RUNX2 gene expression. Future perspective. Recent advancement in science and technology and advanced research on plant extracts is bringing into light their importance in regenerative and therapeutic medicine. As we know very less about the exact site and mechanism of action and side effect of the use of plant extracts, extensive research on humans will help to replace synthetic pharmaceutical drugs to treat diseases. If protocols for proliferation and differentiation of stem cells into desired lineage cells by use of plant extracts can be established, it will help to treat many untreatable diseases like aplastic anemia, leukemia, bone diseases, and cardiovascular diseases. MSCs have promising roles in regenerative therapy due to their broader differentiation potential. From the last few decades, scientists have been aiming to use MSCs for tissue regeneration in bone injury, cartilage injury, spinal cord injury, graft versus host disease, Crohn's disease, and hematopoietic cell recruitment. Though very less side effects of plant extracts on humans have been noted, they may still show adverse drug effects for certain medical conditions which are not well known. With better knowledge of the effects of plant extracts, we may also be able to restrict their undesirable use under certain circumstances. The therapeutic doses can also be well established to have desired effects as well as control toxic effects. Medicinal plants are being widely accepted and increasingly used by the general public for treatment. They are also used as complementary supplements to reduce the side effects produced by Western medicine. The bioactive compounds derived from plants have shown to be potential candidates to activate stem cells for proliferation and differentiation. Currently, recombinant and synthetic cytokines, growth factors, and other proteins are being produced by using bacterial cell, plant cells, and mammalian cells for stem cell growth supplement. These compounds have significant side effects and lead to neoplastic cell transformations with high cost, less stability, and limited application and requiring continuous use, making them unaffordable for low-income countries. Certain medicinal plants have always been grown and used as cultural values for primary health benefits. With more knowledge on values of commonly available plants in the community, it will help people to preserve and use them for healthy living and preventive and curative medicine, and also restrict undesirable use. This will decrease the healthcare economic burden for primary healthcare problems. Thus, plant-derived compounds will be proven as promising agents for stem cell therapy for public health with easy availability and affordability and least or no side effects. 7. Conclusion. MSCs along with medicinal plant extracts have a potential hope in stem cell and regenerative therapy. Plant extracts as stimulants significantly affect proliferation and differentiation into multilineage cells. Bioactive compounds from plants precisely regulate the MSCs through different protein pathways. Medicinal plants/herbs produce less toxic effects, are affordable, and can help to increase disease-treating capability using MSC cell therapy for both non-infectious and infectious diseases. With continued research, by using medicinal plant extracts, improved proliferation and differentiation potential of MSCs will be achieved in the near future, and development of cost-effective technology for cellular therapy will be possible.